Welcome to another lecture in Bio 120. Now we're going to be looking at how to design a growth medium and what are the environmental factors that affect cell growth. This is going to be complementary to the lecture that we saw today, Tuesday, on the growth curve of microorganisms. So let's just begin by how microorganisms are classified according to the energy classes that they use. So we can categorize microorganisms according to the metabolic way in which they obtain carbon, energy, as well as electrons. So when in regard to carbon sources, we can have two categories. Autotrophs are organisms that use CO2 as the sole principal carbon source, whereas heterotrophs are organisms that are going to use a reduced preformed organic molecule such as glucose. Now, the energy that the cells are going to use can come from two different sources. Phototrophs are going to use light, versus chemotrophs are going to oxidize an organic or an inorganic molecule to obtain energy. Last but not least, we have where the organisms are obtaining their electrons. Lithotrophs are going to obtain electrons from reduced inorganic molecules, whereas organotrophs are going to receive their electrons from organic molecules. So when you can see here in the illustration on the right is that the illustration is showing that you have energy sources, either a chemical source for energy or light. If it's going to be light, it's going to be an organism that is going to have phototrophy, and we're going to call that a phototroph. And from there, you're going to have ATP production. So think of any organism that has photosynthesis is most likely going to be phototrophic. Now, if the organism uses a chemical molecule to obtain their energy, that molecule could be either organic, like glucose, or inorganic, such as, for example, molecular hydrogen, or iron, or an amine group. So the organic molecules are going to give you chemoorganotrophs, versus the inorganic molecules are going to be categorized as chemolithotrophs. So in that regard, when we take a look at different categorizations of it, you can see in this table 5.2 from a different textbook that the name of the organisms is going to go in the order of the energy source, followed by the electron donor, followed by the carbon source, and attach at the end the word troph or trophy. So for example, autotrophs is the name of an organism that is going to use light energy, an inorganic source of electrons, and is going to use CO2 as the carbon source. So this table, if you take a moment to look at it, I will give you some examples of an organism that is photolithotrophic. This is the one that we just talked about, which again is going to use light as energy, an inorganic molecule to donate electrons, probably hydrogen for example, and CO2 as the carbon source. And any photosynthetic organism can fall into this category. On the other hand, if you look at a chemoorganoheterotroph, that is an organism that uses a chemical source, an organic chemical source as energy, so for example glucose, that molecule also will provide the electrons as well as the carbon. So here, most non-photosynthetic bacteria, for example E. coli, will fall in that category. So make sure that you understand those three groups of bacteria to be able to then determine what is the energy, electron, and carbon sources. So now, when we look at microorganisms, we're going to look at their nutrients, and we can divide the nutrients as macronutrients, which are required in large amounts, and those are going to be the nutrients that provide carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur. So basically the big macromolecules that organisms are exposed to: carbohydrates, lipids, proteins, and nucleic acids. Now we also are going to know, as you are aware, that organisms are going to require fairly large quantities of ions, for example, potassium, magnesium, calcium, and sodium. And those are going to be coming from salts. So they're going to be present in the cell and are going to be used to help the macroorganisms in their life cycles. Iron is another molecule that is a major macronutrient because of its role in cellular respiration. So let me just take a little moment to concentrate a little bit more about iron, as it's one of the molecules that we really haven't studied a lot, for example, in Bio 110. So as you know, iron, it's present as iron sulfate complexes in many of the proteins found in respiration. It is also the main ion in heme, which, as you know, carries is a prosthetic group, that is going to receive electrons in the electron transport chain. So these iron-sulfur proteins, which could be either found, as a four atom molecule with sulfur, like this one here, 2 irons and 2 sulfurs, or as a cube shown over here in this other form. They're required for passing electrons during the electron transport chain. So iron, it's not an ion that is available very readily in the environment. So microorganisms have devised ways in which to capture iron for the cell, and that is done through molecules called siderophores. So siderophores are small ferric iron chelators. Basically, they're going to have a really high affinity for ferric iron. And ferric iron, as you know, it's an Rs plus 3. So now, these are secondary metabolites. They are amino acid-based molecules which were not made by ribosomal peptides. And they have really high specificity for iron and high affinity for it, and are able to sometimes remove iron from transferrin, for example. Now, the biosynthesis and the uptake of the siderophores inside the cell is regulated under an iron-regulated operon, which we're not going to have the chance to discuss right now. But basically, when the cell is in needs of iron, the iron-binding proteins that are going to sense the amount of iron are going to be made. The siderophores then are going to be generated, and those are going to be released to the cell's environment to capture iron. And then they're going to be brought inside. So the siderophores are going to be made when the cell is low on iron. And the siderophore iron complexes, when it's bound, is going to be bound by a protein that has been termed in E. coli Fab-A. And Fab-A is then able to bring the siderophore iron complex to an ABC transporter. So the Fab-A is an example of the ligand-binding proteins that ABC transporters have. And therefore, at that point, the siderophore iron is able to be transported inside the cell, where it's going to then be reduced and therefore now become ferrous iron, and it will fall off the siderophore. So this image over here, it's showing you a part of the siderophore molecule that actually binds iron, and that molecule is called hydroxamate. The hydroxamate group has a nitrogen that is bound to a hydroxyl group, as well as the carbonyl group over here, and together the oxygen from the hydroxyl group and the oxygen from this carboxyl group are able to bind to ferric iron. So when the siderophore binds to the ferric iron, it's going to be transported across the membrane by the ABC transporter and then reduced in the cytoplasm, and the siderophore is able to be recycled back outside, where it can capture more iron, where the iron that now it's formed in this ferrous state is going to now be used in iron-containing enzymes by the bacteria. So these are two examples of siderophores found in fungi. And what I want to show you is that they are complex molecules that have the capacity to bind iron. For example, here at the top, we have ferrichrome, which is a fungal macromolecule. It's composed of 3 glycines, shown here in green, and 3 modified ornithines, which you have learned already about. But what I did is to give you an idea, it's to mark them in orange for you. So as you see here is the amino and carboxy part of the ornithine and the alpha carbon, and this long chain containing 3 carbons that binds to the nitrogen of what eventually will become the hydroxamate molecule. It's part of the ornithine. So ornithine is a molecule derived from arginine. After arginine has been removed, this group over here that contains these 2 amine groups, that group will be removed to generate an urea molecule and the ornithine. So you can see now that it has an NH3 group over there at the end, and later by different protein modifications, that part will be changed by hydroxamate. Now this other molecule, it's thioacetylfuranine, and is another fungal siderophore that, as you can see, has also hydroxamate groups that are able to capture iron. So iron is going to be one of the very important molecules for the cell to get, especially if those cells are going to be performing respiration. Now, this poses an interesting conundrum because bacteria that are living in the ocean also need to capture iron, but they cannot release acid therefore outside of the cell and expect to keep it, as they're in an aquatic environment and the molecule will be diluted away by diffusion. So what they have resolved to do, and for example in the case of aqua killing, which is shown over here in this image, it's to have a hydrophobic tail that is going to keep the aqua killing molecule anchored to the membrane. And as you can see from this illustration, the aqua killing molecule, it will be exposed to the outside part of the membrane where the bacteria is now going to be able to bind to iron molecules by the hydroxymate groups shown here in blue. But I also put some arrows in this molecule to show you that the hydroxymate group is not the only one capable of interacting with iron. And here, the carboxyl and the amine group and the hydroxyl group, that are present in the backbone of the aqua killings are able to also engage iron and bind to it, probably with different affinity. So what I'm showing on this issue is how a marine microorganism is able to then capture the iron ions. Now, let's take a look at other nutrients. So other nutrients are going to be considered the micronutrients, or we also call them trace elements. These are elements that are required in very small quantities and include manganese, zinc, cobalt, molybdenum, nickel, copper, chromium, selenium, and vanadium, and I forgot what the W is, tungsten, I think it is. Yes, there we go. So what we look over here, for example, is a molecule of molybdenum nitrogenase. And the molybdenum nitrogenase is found in all the bacteria that are going to be able to fix nitrogen to bring it into the biological cycle from nitrogen gas. As you can appreciate, it is also an iron sulfur containing group. But you have a molybdenum ion that sometimes in other variations of this molecule changes to a vanadium, but it is bound together to a homocitrate. And this is part of the enzyme that is going to be able to break nitrogen into nitrite to be absorbed during the nitrogen cycle. And we'll talk about this molecule more detail when we look at the nitrogen cycle by microorganisms. So this is just to give you an example of one of the ways in which these micronutrients are going to be forming part of enzymes and cofactors that are required for the microorganism's biosynthesis. So now in this table 3.1, it's showing you some of these other molecules, and I want you to just take a good look at where some of these ions and elements are going to be used. For example, boron, it's part of the current sensing of many bacteria, or molybdenum, part of the nitrogenase, or then tungsten, which is form of the formate dehydrogenases. So take a look to closely examine how some of these trace elements are going to be utilized even in low quantities for the biosynthetic and biochemical reactions happening in the microorganisms. So they still need to get them from their environment and able to perform the biological functions. Now, growth factors are another nutrient that's required for the organism. So these are going to be essentials for components on precursors that cannot be synthesized by the cell. And oftentimes, some of these molecules are able to be made directly from a microorganism, because these organisms, when you supply them with the mineral source, a carbon source, nitrogen, phosphate, and sulfur, they're able to grow because they contain all the enzymes and pathways that can make every single component of life. As we mentioned in lecture, those are called prototrophs. But oftentimes, a microorganism may lack the capacity to make one of those molecules, and therefore these growth factors come into play because the microorganism now doesn't have the enzymes that are able to make those molecules. So some of the growth factors could be amino acids, some of them are going to be purines or pyrimidines, or even vitamins. So therefore, as growth factors goes, these need to be provided directly to the cell. So what we mentioned earlier in class is that an E. coli can grow in media that contains glucose and salts because it has all the genes required to make every single molecule that it uses. But we're going, for example, to look at leuconostocs which is a microorganism that cannot make the majority of the amino acids that it uses. So therefore, it needs to bring them out from the environment. And it's going to use a transport system that we discussed earlier this week to bring those molecules inside. So here again, the part 2 of table 3.1, it shows some of those growth factors that are not amino acids. So here's showing, for example, folic acid, biotin, vitamin B12, B1, B6, etc., that are picked up from the environment by the microorganisms and most likely transported by one of the many transport systems that we discussed earlier. So we already talked about the fact that an organism is going to require a carbon source, an energy source, and an electron source. And sometimes that could be the same molecule. For example, glucose to E. coli provides carbon, energy, and electrons. Other times those molecules are going to be different. Now we're going to look at how the media is going to be supplemented and what kinds of media you are going to provide for cells that could actually grow in the more minimal sense with only sugar and salt, or when they're required in complex culture media that are going to provide most of the nutrients that the organism needs. So now let's take a look and see how we have two different classes of media. In general, we can divide the media as defined media, in which every component you know by composition and quantity. So in the lab, you get a recipe, you can measure every component for the media to have. So how much glucose it needs, how much concentration of sodium chloride, magnesium sulfate, etc., that are going to provide the salt. And we use those widely in research because we are able to then differentiate what are the nutritional needs of a microorganism and just provide them with the molecules that it needs to grow. On the other hand, we have the opposite, which is the complex media. In the complex media, the exact composition of the components is not known. So they are very rich media that are able to completely satisfy the nutritional requirements of even the most fastidious organisms. For example, oftentimes they contain peptones, and peptones are protein hydrolytes of digested meat, digested soya meal, gelatin, or other protein sources. One of the main components, for example, of a complex media is going to be yeast extract. So you take the yeast, you desiccate the yeast, you grind it, and then you add that to your media. So the yeast is providing all the nucleic acids, all the proteins, all the carbohydrates, and the lipids, but you don't know how much of them are being provided in the yeast extract. You just give it to the cell, and you expect then the cell to be able to break those down and import the monomers inside. So let's take an example of two different kinds of media, and this is table 3.2. For example, here in the left, it's looking at a defined culture media for E. coli. As you can see, it has salts, glucose, and trace elements, and water. So it's completely clear and already put to pH 7. So a salt that can provide potassium and phosphate, one that provides amino and sulfur, one magnesium and sulfur, calcium chloride. There you go. That's all the cell needs. Then the trace elements, also in the form of some kind of salt, also added. So salt and sugar, water, pH 2, pH 7, and E. coli will be happily able to grow. On the other hand, look at the complex media. The complex media is going to have glucose, the yeast extract that I talked to you about, peptone digest, and some salt and water. So the media is brightly yellow. That's probably because of the context that it has. And the microorganism is able to grow. Now, let's talk about Leuconostoc mesenteroides, which is a very fastidious organism, and it lacks the majority of genes that are required to make some of the molecules inside of it. So again, the defined culture media for Leuconostoc is going to have the salts and the carbon source as glucose, but look, it has also sodium acetate. You're providing it with alanine, arginine, asparagine, aspartate, all the amino acids that it cannot make. You're also providing it with the purines and pyrimidines and their precursors so it can make the nucleic acids. And you're giving them a whole vitamin pill to provide all the vitamins that it cannot make. And that is the defined culture media for Leuconostocs because it cannot make amino acids, purines and pyrimidines, and synthesize its own vitamins. Whereas E. coli has all the genes required to make every polymer and macromolecule in its repertoire, Leuconostocs cannot have the genes to make those monomers to then later be able to make the polymers. So those are two examples of a defined media and a complex media. Now, you can also have other classifications for media. Some of them are going to be selective. Why? Because by having a particular ingredient in it, it's going to favor the growth of a particular organism. For example, if you add bile salts or you add crystal violet to your media, that media now is going to prevent the growth of gram-positive bacteria, but is going therefore to favor the gram-negative bacteria to grow. So you can select only for gram-negative microorganisms. On the other hand, you have differential media, and the differential media is going to take advantage of a biochemical pathway that is going to let you tell, well, this organism is able to use this media and have a result, or have a different result in the same biochemical test. So you can distinguish between two bacteria, and it helps you tentatively identify an organism based on the biochemical characteristic. For example, let's take a look at McConkey agar. McConkey agar is a complex medium because it is made with pancreatic digest of gelatin and casein, peptic digest of animal tissue, and everything that has you added to the McConkey agar. But it's also a selective medium because it contains the bile salts and crystal violet, which are going to prevent the growth of gram-positive organisms and only allow gram-negative organisms to grow. Also, it's a differential media because it has lactose as a carbon source, so bacteria which are able to use lactose are going to turn pink in color, whereas bacteria that can grow in there from the peptic digest will grow white when they are not able to digest lactose. So here in the right you have two examples of such bacteria. Here's Serratia marcescens, and it's a gram-negative rod, and therefore look at how gorgeous the red colonies are. And at the bottom you have Shigella flexneri, also showing some of a pinkish looking color because they're able to use lactose. So again, a media could be all the above. It could be complex, it could be selective and differential depending on the ingredients that it has. So in summary, when we think of a media, the culture media is going to supply the nutritional needs of the microorganism, even the most fastidious ones. And you can either have it chemically defined or undefined, as we call complex. Therefore, you also have the issue of a media being selective, differential, or enriched. And those terms describe the media used in the isolation of a particular species or for comparative studies in microorganisms. So now, I'm going to switch gears to talk about some of the environmental factors that are going to affect microbial growth. So now, we're going to start with temperature. So temperature, as you're aware, varies immensely from environment to environment in our planet. And as you know, it affects the rate of enzyme catalyzed reactions. So higher temperature is going to accelerate enzyme catalyzed reactions. Lower temperature is going to slow down those enzyme catalyzed reactions. So, for example, cells that are grown at low temperatures are going to have the slow reaction velocities, but the membrane also can solidify as the membrane loses fluidity. When the increase in temperature happens, we think that the reaction velocity doubles for every 10 degrees Celsius that the temperature rises. So the organism is going to be growing a little bit faster. But at a very high temperature can also slow down growth and be lethal, because now you're having issues of denaturation of proteins, the inability of the organism to repair DNA damage or damage to itself, and then also the fact that now you have the lipid bilayer melting apart and disintegrating. So how does a cell tackles the different temperatures that are faced in their environment? And this is where I'm going to introduce to you the concept of the cardinal temperatures. So the cardinal temperatures describe three different temperatures for an organism: a minimum temperature, the optimum temperature, and the maximum temperature. The minimum temperature is the lowest temperature that the organism can grow. Below that, the organism is going to gel. The transport of nutrients goes so slow that it's incompatible with cell growth. As the temperature increases, you have more speedy enzymatic reactions that are going to now allow the microorganism to finally get to the optimum temperature, where all the enzymatic reactions are occurring at the maximum possible rate. But immediately after that, if the temperature increases, you go to the maximum temperature of life for that organism. And beyond that, the protein are going to be denatured. The cytoplasmic membrane is going to collapse, and the cell will die of thermal lysis. So the cardinal temperatures could be affected by pH and nutrient availability. And as you may imagine, because we have so many different temperatures in our planet, we're going to have organisms that are able to thrive with different cardinal temperatures in those environments. For example, saprophytes, like Polaromas vaculata, are organisms that grow with an optimal temperature of 4 degrees. They can grow at temperatures lower than 0 degrees, but not grow any better at temperatures that pass about 12 degrees. Then we have mesophiles, an example E. coli. And mesophiles are organisms that grow at an ideal temperature of 39 degrees, but as you can see, they can grow pretty well in the 20s and 30 degrees Celsius. But then, reaching past 45 degrees, those organisms have trouble and not grow anymore. And here is when the temperature increases to three categories that include the thermophiles, hyperthermophiles, and very high hyperthermophiles. So as the name implies, a thermophile organism likes the heat. So some of them are going to have an optimal temperature of 60 degrees, other ones 88 degrees, like Thermococcus celer, and other ones even much higher of 106 degrees, like Pherobolus fumari. So the organism doesn't simply tolerate that temperature. It actually requires that temperature to thrive. And all the biological molecules inside that organism has adapted to live at the particular temperature that they're exposed. And that is something that we're going to discuss in a moment. So for example, let's take a saprophyte, which are going to be organisms loving cold environments. So their enzymes, their transport systems, and the protein synthesis all function well at low temperatures. For example, the proteins are going to have higher amount of alpha helices and lesser amounts of beta sheets, which are going to make that protein be more flexible even at low temperatures. They're going to have higher polar groups and lesser hydrophobic amino acids in their molecule, which also includes flexibility. They're going to have lower levels of weak bonds. And now the cell membrane is going to contain a lot of unsaturated or polyunsaturated fatty acids. And as you remember from Bio 110, those are going to remain semi-fluid when cold. So if you bring up the temperature for the saprophytes above 20 degrees, now they start to have membrane failure and they're going to leak cellular components into their environment, so they die. On the other hand, we have psychotrophs, which are microorganisms that can grow between 0 to 7 degrees, but their optimum is about 20 to 30. One example that all of us have encountered is mold, and the mold grows in the bread that we put in the fridge. So a lot of bacteria and fungi are responsible for food spoilage fall within the psychotrophs because they're going to be the ones growing in the experiments that we keep in our refrigerator. On the other extremes, we have the thermophilic bacteria. And the enzymes, their transport system and protein synthesis mechanisms all are going to function and require that high temperature. So their proteins are very heat stable. And they have a lot of organized hydrophobic interiors that are going to keep them nice and tight and they will have to have really high temperatures to actually become denatured. For example, they're going to have a lot more hydrogen and non-covalent bonds that are going to strengthen the structure so they don't fall apart that easily. You have prolines that make polypeptide chains less flexible so they don't vibrate as much. And the proteins are stabilized and folded often by the presence of chaperones, which are going to keep them in a stable conformation. Now, the cell membranes are going to have high level of saturated and branch fatty acids that are going to remain rigid when hot, maintaining the fluidity of the membrane compatible with the temperature. So for example, some archaea that, as you know, contain lipid monolayers made of isoprene polymers. Those are able to resist really high temperatures because instead of having a bilayer that can fall apart, now have a monolayer that can resist thermolysis. So this table 5.1, shows the known upper limits basically of the temperature that organisms can grow. For example, insects can be happily growing at 45 to 50 degrees Celsius. Whereas when we look now at microorganisms like algae, they can actually sustain temperatures of 55 to 60 degrees. But the champions of thermophilic living are basically microorganisms, particularly bacteria, and more specifically archaea. So you have some anoxygenous prototrophs that are able to live happily at 70 to 73 degrees Celsius, other ones at 95 degrees Celsius. And archaea, which are extremophiles, can live, especially if they're chemoorganotrophs or chemolithotrophs, they can live in temperatures of 122 degrees. Imagine putting your hand in water that is 122 degrees Celsius. You will literally lose the skin of your hand. But these microorganisms, they have evolved protein and membrane structures that are compatible with living at those temperatures. Now, let's take a different turn and look about pH. And as you know, pH is the measure of hadronium ions or protons in a solution. And you define pH as the negative log of the concentration of protons or hadronium ions. Either two words. Now, cells are going to maintain a pH in the cytoplasm of about 7 pH. So if you have variations in pH, if you put an organism in different pH, that can disrupt the plasma membrane. Enzymes are not going to function anymore because they're going to fall apart and be denatured. And of course, that can affect membrane transport. And any organism will die if the internal pH of the cytoplasm drops below 5.0. So how are organisms then able to live in such a great range of pH? So we're going to look at organisms that like living in acidic environments, we call them acidophiles. And organisms that love living in alkaline environments are called alkalophiles. So some organisms, for example, Helicobacter pylori, lives happily in the lining of our stomach, which has a pH of between 1 and 2, versus other organisms are living in alkaline soda lakes that are at pH of 12. But they have to find ways in which they can now conquer those different environmental pH changes. So for example, of acidophiles, the optimum pH to grow is between 0 and 5.5, so really acidic pHs. Neutrophiles grow between 5.5 and 8, and alkalophiles are going to grow in pH of 8.5 to 11.5, very alkaline environments. But we had said that the cytoplasmic pH needs to maintain neutral, around 7, and therefore the plasma membrane has to maintain its impermeability to protons one way or another. So in that case, neutrophiles can exchange, for example, potassium for protons using an antiporous system. So they can then pump the protons out of the cytoplasm and bring in potassium ions, and therefore maintain the pH of the cytoplasm constant. Alkalophiles, on the other hand, are going to maintain a neutral pH because now they're able to exchange internal sodium for external protons. So basically bringing protons from the outside and exchanging them from internal sodium. And they also have other ways in which they can buffer their cytoplasm. So as I mentioned, a unified feature of bacteria is that the cytoplasmic pH is always about 7. And acidophiles and alkalophiles both have strategies that are going to maintain the pH in the cytoplasm around 7. So they can counterbalance entry of protons by proton pumping them out or uptaking potassium, or they can buffer their cytoplasm because they can synthesize glutamate or polyamines, and therefore maintain their pH inside the cytoplasm as neutral as possible. So what can they do? They either can have potassium or sodium proton antiporters that are going to correct small changes in pH, but those are not fast enough to remedy a very drastic change on pH. So for example, E. coli and Salmonella typhimurium have what is called an acidic tolerance response, where they are going to be able to use a proton translocated ATPase to contribute by making more ATP or pumping out protons. So those proteins that are going to be part of that acidic tolerance response are going to be activated when the pH, for example, drops below a particular threshold. Also, the microorganisms can make chaperones that are going to resist acid or heat shock, like the ones that we have talked before, and therefore prevent the denaturation of the proteins when the pH changes drastically. Now, as a consequence of pH, we have also the term of osmolarity, that, as you know, is the capacity of the membrane to be permeable to water. So the water availability, we're going to talk about that. The amount of water that can cross the membrane will depend on the amount of solute that is present in or outside of the cell. So, for example, in a hypotonic environment, water will enter the cell and cause it to swell until it bursts. And the cell will prevent that bursting if something can be done to prevent the influx of those water molecules. So what happens is, as we discussed before, remember, many microorganisms have inclusion bodies, and the inclusion bodies can be considered a solute, and therefore, when you increase the number of inclusion bodies inside the cell, that reduces the osmotic concentration in the cytoplasm, and that helps tie up molecules in a particular form. Cells also have mechanosensitive channels that are going to open up and release water molecules when the plasma membrane expands and stretches to allow the channel to open up. So it's sort of like a pressure gauge that is present on the membrane. But even at certain concentrations, those mechanosensitive channels and the changing in solutes are not always capable of helping the cell when the osmolarity of the solution changes. So this is when we talk about water activity. And water activity, it's a measurement of how much water is available to a cell in a particular environment. All the environments that we think about are aqueous in one way or another. And the water activity is then defined as the ratio of the solution's vapor pressure to the vapor pressure of pure water. So if you take a solution of pure water, the ratio of the vapor pressure of water vapor to the vapor pressure of pure water is going to be 1. And most microorganisms are going to be happily living in water with that kind of water activity. But now, as the amount of solute changes, like in the case of seawater or maple syrup, or, for example, a salami, now the ratio of the vapor pressure to the vapor pressure of water changes and diminishes, and that makes an environment having less water availability. We often use that in the processing of food, like in the case of cold cuts that have been dry, like salami, or jams where now you have increased the concentration of sugar, or in salted animals like salted fish or candied fruit, we now reduce the water availability, which is now going to be detrimental for the cell, because as the solute inside the cell is less than the outside, the water is going to rush out of the cell and make them dry up. So very few organisms are osmotolerant. And for example, Halobacterium are bacteria that are able to grow in super salt lakes because they have mechanisms to control the osmolarity and therefore not swell when the water activity is reduced to 0.75. So let me take a look at the issue of compatible solutes. And compatible solute is a way for cells to now change the solute concentration in the cytoplasm so they don't suffer osmotic lysis. So some of these compatible solutes are called compatible because they are quote-unquote compatible with metabolism and cell growth. Some of them are going to be molecules like choline, betaine, the amino acid proline, the amino acid glutamate, or another amino acid that is made inside the cell and kept at a particular concentration, so the solute concentration is high enough so the water doesn't want to rush inside the cell. Other times, this is done by elevating the potassium levels inside the cell. And carbohydrates like sucrose or some polyols like arabinol, glycerol, or mannitol are used in algae and fungi to also increase the solute concentration inside the cell. So those microorganisms living in low water availability usually make compatible solutes so they do not lose their water to the environment. And here, table 5.4 shows some of these examples. So I will leave the table here for you so you can see the comparison of some of these compatible solutes. So for example, you have amino acids like glutamate or proline that are going to help microorganisms living in a water availability of 0.9 to 0.98. But when you're going down to water availability levels of 0.75, now, for example, some of the halophilic green alga, Dunaliena, are going to make glycerol to maintain the concentration of solute high inside the cell, and therefore they don't lose their water to the environment. So this is just an example of some of those compatible solutes. One word that I want to able to bring to you, it's the xerophile. And the xerophiles are microorganisms that are able to live in low water activity conditions. Which brings me to the halophiles, which are microorganisms that grow in the presence of high concentrations of sodium chloride or salts. Now, some of them are going to grow at much higher levels. So the extreme halophiles, like Halobacterium salinarum, are able to thrive in sodium concentrations of 6.2 molar. Now, as I mentioned to you, they also have a similar table to the cardinal temperatures, but in this case it's going to be the cardinal concentration of sodium chloride. So most non-halophiles, like E. coli, can only grow at a very narrow concentration of salts, where you have some halotolerant bacteria like Staph aureus, which are able to grow at some concentrations of salt that are a little bit high, but they don't like it very much. Now, some really true halophiles, like the marine microorganism Vibrio fischeri, definitely love to live at a much higher concentration. But it is not until you get to the extreme halophiles, like Halobacterium salinarum, which lives in the water of the Salt Lake in Utah, that you get to bacteria that really thrives in the environments that are super high concentration of salt. So these microorganisms that are extreme halophiles have modified the structure of their proteins and membranes, and rather than just simply increase the compatible solute concentrations. And again, they require those really high sodium concentrations and chlorine concentrations to make sure that their structures are stable. For example, they accumulate a very high amount of potassium inside so they can stay hypertonic as compared to their environment. So really high concentration of potassium. Because of that high concentration of potassium, their enzymes, the ribosomes, and the transport protein all are going to require high concentration of potassium. Now, their membranes on the outside are stabilized by high sodium concentrations because that area of the cell is exposed to that high concentration of sodium. So they have the high concentration of sodium in the outside that is going to stabilize the membrane. They have a high concentration of potassium in the inside, and therefore all their enzymes and molecules require and thrive in that high potassium concentration. Last but not least, I'm going to talk to you about oxygen. And here we have 5 different microorganisms categorized according to how much they like oxygen. So in the left, we have 5 different tubes, and those different tubes contain thioglycolate broth, which is a kind of media that had been spiked with some agar to make it a little bit viscous. They also added the red dye resazurin, and that dye turns pink when there's oxidation, so only in the area where oxygen is present. So now you have bacteria that are aerobic, like Pseudomonas aeruginosa. They require oxygen to live. And as you can see in part A of the figure, those microorganisms are mostly growing in colonies at the oxygen-oxic interface. Very few, if any, are growing in the anoxic region shown in the yellow area. Now, anaerobic bacteria are bacteria that cannot live in the presence of oxygen. And we're going to see why in a moment. Oftentimes, oxygen is toxic to them. And therefore, as you can see, most of the colonies in the experiment are showing the bacteria thriving in the farthest location away from the oxygen. Now, other microorganisms are going to be facultative aerobes. E. coli is one of these. Therefore, the microorganisms are going to grow really well in the presence of oxygen. When oxygen concentrations are low, they now switch to fermentation and are able to grow as well. So, facultative aerobes are able to respire when oxygen is present, but ferment when oxygen is not present. Yeast, for example, is a facultative aerobe. You have other bacteria which are called microaerophilic, and the microaerophilic bacteria need oxygen, but not at very high concentrations. And last, you have the aerotolerant anaerobes. Those are bacteria that oxygen doesn't damage them, but the bacteria only has the capacity to ferment, and therefore cannot respire. And those are the ones shown in E. Now, this table, table 5.5, it's showing you some of the kinds of relationship with oxygen to the microorganisms. So an obligate aerobe, it's going to require oxygen because it can only respire. And you, of course, are going to have them in places where oxygen is present, like our skin. Whereas a facultative aerobe does not require oxygen, but it grows better if oxygen is present, like E. coli. So they can grow by respiration if air is present, but they can grow either by anaerobic respiration or by fermentation when oxygen is not there. And that will be E. coli. And then microaerophilic bacteria require oxygen but at much lower concentrations. They usually respire, and Spirillum volutans is one example. And they live in the lake at depth where oxygen levels are not that high. Now the aerotolerant bacteria, as I mentioned to you, do not require oxygen, and it doesn't matter that oxygen is present, so they can grow by fermentation anywhere. And that Streptococcus pyogenes is a good example. But then you have the anaerobic bacteria that are obligate anaerobes because oxygen is harmful or lethal to them. They only grow by either fermentation or anaerobic respiration, and Methanobacterium forminicum, for example, is a type of bacteria that is an obligate anaerobe. They often are living in very oxygen-deprived environments. So why is oxygen toxic to some microorganisms? Well, as you may imagine, oxygen is used in respiration. But oxygen itself, the O2 molecule, it's not toxic, but it's the byproduct of the reduction of oxygen to water that now it's toxic. So you have the superoxide ion, you have hydrogen peroxide, you have the hydroxyl radical. All those three are reactive oxygen species. And they can be generated even in a cell that is not using oxygen because they have haploproteins or quinones, or sometimes iron sulfur proteins that catalyze these reactions and generate these molecules. And this figure 5.29, it's showing you the steps in which you have an oxygen molecule being completely reduced into water and how some of those intermediate reactive oxygen species are generated. Now, traditionally, cells have ways to defend themselves against these molecules, and we call them antioxidants. So when a cell is living in an environment that contains oxygen, that means that the cell has to have a way to defend itself from those toxic oxygen reactive species. One way in which bacteria does that is by having enzymes such as catalase, peroxidase, or desuperoxidase dismutase, or SOD. So, for example, the enzyme catalase is going to be able to take hydrogen peroxide and now convert it into water in oxygen. And as you can see in figure 5.31 shown here, one of the tests that we do is the catalase test. So you can put a culture and you add hydrogen peroxide, and now you're going to get very vigorous bubbling, and that bubbling is the result of the oxygen being generated. That's why hydrogen peroxide, we use it as a disinfectant, because it's going to help kill microorganisms, preferably those that cannot have catalase, or the catalase system could be overwritten by the hydrogen peroxide that you're giving it to disinfect. Peroxidase also eliminates hydrogen peroxide, now by using also NADH, and that's going to happen now, generate water molecules, but an NAD+. Now, the superoxide ion, it's a little bit more tricky to be eliminated because the enzyme that has that job, superoxide dismutase, is going to take two superoxide molecules with two protons and convert that into a hydrogen peroxide, an oxygen molecule. So the cell now, if it's able to get rid of the dangerous superoxide, now has to deal with the hydrogen peroxide generated by superoxide dismutase. That's why superoxide dismutase often works together with catalase to get rid of the superoxide ions. So here in number 4, in the purple square, you can see that. So I believe that's the last slide that I have in the lecture, so I'm going to stop there. And I'm going to post that for you today and be ready to have a quiz. So if you have any questions, please post us online. Thank you very much, and I'll see you on Thursday.